Today's topic is cerebral physiology and its applied aspects.
We will study the topic under following headings. Firstly, anatomy of cerebral circulation. Anterior circulation is formed by pair right and left internal carotid arteries. Posterior circulation is formed by right and left vertebral arteries. The internal carotid arteries and the basilar artery, formed by connection of two vertebral arteries, connect to form a vascular loop called circle of Willis. Here is an pictorial representation of circle of Willis. As we can see, circle of Willis starts with vertebral artery, which gives branches like anterior spinal artery and the posterior inferior cerebral artery. The vertebral artery combines to form basilar artery. Basilar artery gives out many branches like anterior inferior cerebellar artery, pontine arteries, labyrinth arteries, etc. etc. Next, after the basilar artery divides into the posterior cerebral arteries, which gives out the posterior communicating branch and the internal carotid branch, which further combines to form anterior communicating artery. Under normal conditions, blood from the anterior and the posterior circulation does not admix because pressure in the two systems are equal. Substantial variability exists in anatomy of circle of Willis, and a significant proportion of individuals may have an incomplete circular loop. The second heading we will study is venous drainage. Superficial cortical veins are within the pia mater on the brain surface. Deep cortical veins drain the deeper structures of the brain. These veins ultimately drain into the dural sinuses, which further drain into the right and left internal jugular veins. In approx. 65% of the points flow in the right internal jugular vein is greater than in the left internal jugular vein. The next topic we will study is cerebrospinal fluid. The total cerebrospinal fluid space is approx 150 milliliter, and total daily CSF production average between 450 ml. CSF is produced primarily by choroid plexus in lateral, third and fourth ventricles. CSF production is also under the influence of circadian rhythm, with peak production during sleep. CSF production is a result of hydrostatic efflux of the capillaries into perivascular space, then active transport into the ventricles. CSF reabsorption occurs primarily by arachnoid granulations present in dural sinuses. Here is a video to explain better how the CSF flow pathway works. CSF is cleared from the circulation by glymphatic pathway. Conceptually, the glymphatic pathway can be visualized as a system akin to the lymphatic system in the systemic circulation. Note, however, the brain does not contain lymphatics other than those present in meninges. Functionally, the CSF enters the periarterial space, a space that is bounded by vessels and the end of the astrocytes. Aquaporin channels on the end feet facilitate this water exchange. From the periarterial space, CSF is transported into the brain parenchyma, and from there to the perivenous space onto the ventricles. The periarterial space increases significantly during speed and during general anesthesia. Hence, lymphatic transport and waste clearance is increased during these states. Here are some applied aspects for CSF. Firstly, PDPH, post-dural puncture headache. PDPH is believed to result from the leakage of CSF from a dural defect, post-spinal anesthesia, and intercranial hypotension. Loss of CSF at a rate faster than it can be produced causes traction on the structure supporting the brain, particularly the meninges, dura, and tentorium. Increased traction on the blood vessels and the cranial nerves may also contribute to the pain. Secondly, meningitis. Infection of the subarachnoid space can follow neural axial as a result of contamination of the equipment or injected solutions, or as a result of organism tract inform the skin. Indwelling catheters may become colonized with organisms that track deep causing infections. Next heading we will study will be cerebral blood flow. The adult human brain weighs approximately 1350 grams and therefore represents approximately 2% of the body weight. However, it receives 12 to 15% of the cardiac output. This high flow rate is a reflection of the brain's high metabolic rate. At rest, brain consumes oxygen at a rate of approximately 3.5 milliliter oxygen per 100 gram brain tissue per minute. Next, the factors influencing the cerebral blood flow. Firstly, myogenic regulation. In normal human subjects, CBF is auto-regulated between 70 mmHg, lower limit of the auto-regulation, and the 150 mmHg, upper limit of the auto-regulation. Above and below the auto-regulatory plateau, the CBF is pressure dependent, pressure passive, and linearly varies with CPP. Second, the chemical regulation of cerebral blood flow. Regional CBF and metabolism are tightly coupled. Increased neuronal activities results in increased local brain metabolism, and this increase in CMR is associated with a proportional change in CBF, referred as neurovascular coupling. The traditional view of neurovascular coupling is that it is a positive feedback mechanism wherein increased neuronal activities results in a demand for energy. This demand is met up by an increase in CBF. More recent data suggests that the coupling is based on a feed-forward mechanism wherein neuronal activity directly increases CBF, thereby increasing energy supply. Functional state. CMR decreases during sleep and increases during sensory stimulation, mental task, or arousal of any cause. Temperature. The CMR decreases by 6 to 7% per degree Celsius of the temperature reduction. Hypothermia can also cause complete suppression of the EEG at approximately 18 to 20 degrees Celsius. This suppression occurs because hypothermia decreases the rate of energy utilization associated with both electrophysiologic function and the basal component related to the maintenance of the cellular integrity. The CMR oxygen at 18 degrees Celsius is less than 10% of the normothermic values, which explains the brain's tolerance for moderate periods of circulatory arrest at these and colder temperatures. Hyperthermia has an opposite influence on the cerebral physiologic functions. Between 37 degrees Celsius and 42 degrees Celsius, CBF and CMR increase. However, 42 degrees Celsius, a dramatic reduction in cerebral oxygen consumption occurs, an indication of threshold for a toxic effect of hyperthermia that may occur as a result of protein denaturation. PaCO2. CBF varies directly with PaCO2, especially within the range of physiologic variation of PaCO2. CBF changes 1 to 2 ml per 100 gram per minute for each 1 millimeter Hg change in PaCO2 around normal PaCO2 values. This response is attenuated at a PaCO2 less than 25 mmHg. Under normal circumstances, the sensitivity of the CBF to changes in PaCO2 is positively correlated with resting levels of CBF. The magnitude of the reduction in CBF caused by hypocapnia is more intense when resting CBF is increased. Conversely, when resting CBF is reduced, the magnitude of the hypocapnia-induced radiation in CBF is decreased slightly. PaO2. Changes in PaO2 from 60 to more than 300 mmHg have little influence on CBF. A reduction in levels below 60 mmHg rapidly increases CBF. Below a PaO2 of 60 mmHg, there is rapid reduction in oxyhemoglobin saturation. The relationship between oxyhemoglobin saturation as evaluated by pulse oximetry and CBF is inversely linear. A reduction in arterial oxygen content and therefore cerebral oxygen delivery can be achieved either by reduction in PaO2, hypoxemic hypoxia, or by reduction in Hb concentration, anemia, hemodilution. Both of these situations lead to cerebral vasodilation and increase in CBF. The rostral ventrolateral medulla serves as an oxygen sensor within the brain. Stimulation of RVM by hypoxia results in increase in CBF, but not the CMR, and the lesions of RVM suppress the magnitude of the CBF response to hypoxia. Effects of blood viscosity. Hematocrit is the single most important determinant of blood viscosity. In healthy humans, variation of the hematocrit within the normal range, 33% to 45%, probably results in only modest alterations in CBF. Beyond this range, changes are more substantial. In anemia, cerebral vascular resistance is reduced and CBF increases. However, this may result not only from the reduction in viscosity but also as a compensatory response to reduced oxygen delivery. Cardiac output. Though not yet completely established, but recent data suggests that cardiac output impacts cerebral perfusion. In several investigations in which central blood volume was modulated, either reduced by application of lower body negative pressure or increased by infusion of fluid, a linear relationship between CO and CBF, as measured as middle cerebral artery flow viscosity (MCA-FV) by transcranial Doppler, was clearly demonstrated.